Hello and welcome to today's American Physiological Society Endocrinology and Metabolism section webinar entitled Understanding Body Temperature and Energy Homeostasis in Mice. I will be serving as your moderator today. My name is Dr. Lizzie Janet Cardoso. I'm a professor at the University of Mississippi Medical Center, a longtime member of the American Physiological Society and the chair of the APS Endocrinology and Metabolism section. In today's session, Dr. Mark Reitman, MD, PhD, will examine strategies for choosing ambient temperature for studying mice and describe how ambient temperature affect metabolic rate and physiology, as well as concepts such as thermoneutrality and zones versus points. A few housekeeping notes. Out of respect for our speaker, we ask that you refrain from photo or video capture of the presentation in whole or in part. This section will be recorded and available for future viewing through a link on the registration website. Taking photograph of another person's presentation without their explicit permission is prohibited. At this time, all attendees except the speaker are in listen-only mode with video off. As moderator, I'll be happy to raise questions that are entered into the Q&A function on the bottom of your screen. Before we begin, I would like to give a special thank to our today's sponsor, Columbus Instrument, for their continued support to the Endocrinology and Metabolism Sections Award and social activities. Thank you so much. The session will end at around 12 p.m. Eastern Time. It's now my pleasure to introduce Dr. Reitman, MD, PhD. He is the chief of the Endocrinology, Diabetes and Obesity branch in the National Institute of Diabetes and Digestive and Kidney Disease at the NIH. His research focused on using mice to understand the physiology of obesity and body temperature, with interest in the neural regulation of metabolic rate and body temperature, the bombensin-like receptor 3 genes, and using mice to forecast drug efficacy in humans. Thank you so much again, Dr. Reynolds, for being with us today, and the floor is yours.
Thank you very much. I'm just turning on my laser pointer. Okay, thank you very much to the APS and to Columbus Instruments for the opportunity to give this webinar. It's really my pleasure. I enjoy doing it, and I hope this is interesting and useful for people. I've added a subtitle to the seminar title that mice are not small humans, which I think you'll recognize by the end of my talk. And again, if anybody has questions and wants to reach out to me personally, my email address is below. Okay, so why study energy expenditure? Why study mice? First of all, I'm a fan that fundamental physiology is really important, and any way we can learn about it is good. And mice are the most studied laboratory mammal, and so it's important to understand them. I think I'll convince you that fundamental differences exist in energy expenditure between mice and humans, and we need to understand how mice energy physiology applies to humans to identify conditions where mice are a better model of human physiology. I spent 9 years at Merck helping discover and develop anti-obesity drugs, and I came away with that realizing that we really didn't understand the physiology of obesity, and that's really one of the things that led me to try and make mice better models. So what I'll do today is I'll review indirect calorimetry, give you a little bit of the theoretical background, introduce some thermal biology, and then concentrate on environmental temperature, body temperature, energy expenditure, and how this impacts drug development for obesity. A very, very short introduction that obesity is a worldwide problem. It affects all parts of the world. It's increasing. And just so people in East Asia and South Asia don't get too complacent, the complications of obesity, whereas obesity is considered a BMI of 30 or greater in Europe and the West, in Japan, obesity is a BMI greater than 25. So obesity is a worldwide problem. And pharmacotherapy for obesity has a long and checkered history. I'd like to just make a few points based on this slide. First of all, that we're trying to make drugs that are going to be long-term therapies. And that means they must be very, very safe, because while obesity does increase the risk for things like cardiovascular morbidity and mortality, it's a modest risk. It's not a huge risk. And if you're going to be taking a drug for the rest of your life, you want to be darn sure it's a good, safe drug. So the goal is for treating obesity is a reduction in the comorbidities. And what's very interesting about it is that many drugs have been developed and some marketed for obesity. And of all these drugs, only one was rationally designed, orlistat. All the rest were accidentally discovered to cause weight loss and therefore developed for obesity, for the treatment of obesity. The other point I want to make is that almost all the compounds and drugs are brain mechanisms. So obesity drugs work in the brain. And the last thing I really want to say is it's been a real revolution in the last few years with now very effective, for the first time, very effective therapy for the treatment of obesity with the development of the GLP-1, glucagon-like peptide-1 agonists and related molecules. So it's really an exciting time in the obesity field to be here. So obesity is fundamentally an imbalance between food intake and energy expenditure. And again, it's all controlled by the brain. But what's interesting is food intake is easy to measure. It's relatively more important than energy expenditure and is really much more studied compared to energy expenditure, which is really one of the reasons I think I've chosen to concentrate on trying to understand the energy expenditure aspects a little bit better, and thus the title of this talk. So what is indirect calorimetry? Indirect calorimetry, you're just measuring energy expenditure, and you do that by measuring oxygen consumption and CO2 production. This is the same as humans or any other animal or species or bacteria. It's fundamental. What you do is you put room air into your system, you measure the O2 and CO2 out, measuring the delta in O2 and CO2. And in mice, you have the advantage that you can put many of them in a chamber at the same time, change the temperature of the chamber, and monitor the activity of the mouse with infrared beam break. We typically, almost all the time, also continually measure core body temperature by telemetry and also measure food intake. And just to be a little more didactic too, what is it, how do you approach the calculation of metabolic rate from the oxygen consumption? And you see that if you burn carbohydrate, exemplified here by glucose, that for every mole of oxygen consumed, you produce 1 mole of CO2, and it's 112 kcals per mole of energy released available for metabolism from that. If instead you burn fatty acids, you get slightly less kcals per mole oxygen consumed, and you have fewer moles of CO2 produced. And the carbohydrate is pretty much true for most carbohydrate sources. Fatty acid varies a little bit based on the fatty acid, but is pretty reasonable. Protein varies a lot, is not a main energy source, and is typically not included in most studies of mouse metabolism. There's more information that you get from indirect calorimetry by focusing not just on the O2 produced, but it's not just the O2 consumed, but the CO2 produced. And you see that you get the respiratory exchange ratio, meaning 6 moles of O2 are consumed per mole of glucose, producing 6 moles of CO2 for an RER of 1. Whereas when you do the same thing for fatty acid, the RER is 0.70. So by understanding the RER, which ranges from about 0.7 to 1, you know algebraically what the fuel being burned is. And you can also more accurately, now you can adjust the exact kcals per mole oxygen to get even a more precise measure of the metabolic rate. So what are some reasons that you have to think differently about mice and humans? And here I just put one example up. We define the basal metabolic rate, which is typically what's reported for humans, is being defined at physical rest. Yet mice do not stop being physically active when you ask them to. BMR is measured in the post-absorptive state, meaning overnight fast. But in mouse, fasting rapidly causes adaptation, such as increased physical activity, followed by a reduced metabolic rate, reduced body temperature, and eventually severely reduced metabolic rate and body temperature and torpor. In mice, we typically do not measure EEGs and EMGs to know if a mouse is awake or asleep when you're measuring it. And finally, I'll go through the fact that humans are measured at thermoneutrality, BMR is measured at thermoneutrality, but mice have very different thermal biology. So mice are to try and report a BMR in a mouse is a really difficult task compared to the routine way it's measured in humans. So my last sort of didactic slide on mouse indirect calorimetry is just a plea that you report the total energy expenditure as a rate per animal. If at all possible, compare cohorts that have the same body weight, compare animals of the same body weight, and never ever divide by body weight. Use ANCOVA if weights differ. And I also really like people to report the RER because it tells you the fuel source. The average RER should approximate the food quotient, which tells you also the food quotient for chow is about 0.91. For a high-fat diet, it's about 0.79. So again, it gives you a chance to make sure that your system's working properly and the RER is biologically plausible. Another just pearl is that I think reporting the VO2 and the VCO2 does not add any information beyond the TEE, the total energy expenditure, and the RER. And so I would just advocate not bothering with the O2 and the CO2, just report the TEE and RER. And finally, at steady state, the energy expenditure should equal the metabolizable food intake. And as a technical check, are the TE and food intake roughly the same? I mean, I've seen multiple examples where there's a twofold difference. So clearly something funny is going on in that publication. Some other details: the calorimetry chamber is a very unusual environment. So you should ideally acclimate the animals for at least a day, and ideally more than that, till they have more normal behavior in the calorimetry chambers. And the other point is that 5% changes in energy expenditure are biologically significant, but difficult to detect. 10% should be pretty detectable. And just parenthetically, most experiments are actually underpowered in the way we do them, unfortunately. So now what I want to switch now is talk about the role of ambient temperature, TA, and body temperature, TB, in calorimetry. And this all is a consequence, the thermal biology difference is a consequence of body size. So for example, a small mammal like a mouse versus a large mammal, we have the same median body temperatures, a similar diurnal rhythm. However, in the small mammal, the body core is very poorly insulated. The animal lives below thermoneutrality. Some heat comes from metabolism, but much more is needed. So there's a large role for brown adipose tissue. And the physiology is oriented toward heat conservation and heat generation. In contrast, in a human, body core is insulated. We adjust our environment so that it's thermoneutral. Most of, or really basically all the heat we need is a metabolic byproduct. So in adult humans, there's little role for any brown adipose tissue, and the physiology of big mammals is oriented toward heat dissipation. The exact ways are often species dependent, but certainly vasodilation and modification of environment and behavior are major. Body temperature is highly and tightly regulated in both big and small mammals, but small mammals have some extra tricks that large mammals don't use. Although the temperature is still highly regulated, it's allowed to vary. And just again, to go through an example of this difference in scaling between mice and humans is that a 25-gram mouse is a 1/3000th the weight of a 75-kilogram human. But if you look and calculate that the surface area for a kilogram of mouse is 14 times the surface area of a kilogram of human, and similarly, the heat loss from a kilogram of mouse is 17 times the heat loss of a kilogram of human. So a mouse is a little radiator. And that is really one of the big reasons for such a difference in energy metabolism, because the mouse has to generate all this energy that it's losing just to stay warm. So this is an example of primary data. What we've done is we've acclimated the mice, put them in indirect calorimetry chambers. Each of these, between each red line is a day. This is the dark phase, light phase, dark phase, light phase. The mice were put at 22 degrees. It was ramped up to 33 and then back down to 4. You see the energy expenditure. It's lower at 33, higher at 4. You have the respiratory exchange ratio showing that there's greater food intake at night and therefore a higher RQ. You see activity is higher in the dark phase and body temperature is higher in the dark phase. And this is the sort of raw data we get and play with. And sort of the classic way of understanding metabolic rate data and the effect of ambient temperature is called a Scholander curve, although I really think it's really presented 16 years earlier by Kleiber. And what I want to do is take you through this curve, because understanding this curve really explains a lot of thermal physiology. It's how people think about it. So the first thing you look, and what we've done here is graphed energy expenditure versus ambient temperature. And the first thing you see is there's a minimum at the basal metabolic rate. If you look, the end of the basal metabolic rate, where the whole region where the body metabolic rate is at minimum, is called the thermoneutral zone. When you get to the lower end of the thermoneutral zone, the critical temperature below which energy expenditure has to be maintained, energy has to be expended to maintain body temperature. And the line you generate is the heat requirement to maintain that body temperature. If you extrapolate that line back to no heat needed, that's the defended body temperature. So here you've never measured body temperature. You need not have measured body temperature, and you can determine what the body temperature set point is. And finally, the slope of this line is the heat conductance, which mathematically is 1 over the slope is the insulation. So by understanding this curve and plugging your data and thoughts into it, you can really learn an awful lot about energy metabolism. And this is what we've taken primary data and put it into this format and divided sort of in a phase diagram for energy expenditure. And you can see here that you have in gray the basal metabolic rate, in green the energy cost of physical activity, in orange brown you have the cost needed to assimilate food, which is called the thermic effect of food. That is the energy increase that occurs after a meal. And finally, everything in blue here is all the energy being expended just to defend body temperature. And since we have typically studied mice at 22 degrees, you see that about a third of energy expenditure by a singly housed mouse at 22 degrees, is devoted to cold-induced thermogenesis. And what's important to recognize at steady state that energy expenditure is the same as energy intake or food intake. So a third of food intake in a single housed mouse at 22 is being burned just to stay warm. That leads to some interesting biology, which is that if you fast a mouse, it can save a lot of energy by letting its body temperature drop. So what we have here is, again, light phase, dark phase, and each of these lines is the core body temperature of an individual mouse. And when you take away the food in the middle of the light phase, you see the mice run around looking for food. They enter the dark phase, then they realize there's no food around, and they go into torpor. And torpor is a large drop in body temperature and metabolic rate that requires only a cool environment. Room temperature is cool, a quiet environment. This works really well on weekends, and inadequate food. And this allows the mouse to save a tremendous amount of energy. And this is normal biology for all mice. This is not a sick mouse. This is good physiology to survive starvation. So this brings up the question, what about humans? Humans are probably somewhere here on the curve where we have a little bit or maybe no cold-induced thermogenesis typically. Can we study mice at a temperature that better models human physiology? So how do we optimize ambient temperature to understand the obesity drug efficacy? And so what we did a bunch of years back is took 2-nitrophenol, which is a chemical uncoupler, which degrades the mitochondrial proton gradient, and studied it in mice at both 22 and 30. So if you treat mice with DNP at 22, you end up with a metabolic rate. I've just graphed this sort of an arbitrary scale, but these are real data. And you see that if you treat with DNP indicated in blue, that now you've reduced the cold-induced thermogenesis in green, but you have the same total metabolic rate. So what happens if you treat with DNP at 22 degrees, you reduce the brown fat capacity, but there's no change in total energy expenditure, no change in food intake, no change in body weight or adiposity. If now instead you do the exact same treatment, but it is now done at 30 degrees, you now see that there again is a reduction in brown fat capacity. Now you see the increase in energy expenditure because you couldn't turn down the cold-induced thermogenesis. And for some reason, there was no increase in food intake, which I had expected. So instead, what you do see is a reduction in body weight. So here you have very different physiology of a peripherally active chemical uncoupler at 22 versus 30 degrees. And so I've given you one example of dinitrophenol. This is a collaboration study with Rune Corey's group at Novo Nordisk. And what we did was summarized all the drugs we could find for mouse at 22 versus 30 and found some drugs like GLP-1 agonists work equally well, FGF21 also, whereas some work much better at 22 than at 30, and some work much better at 30 than 22. But the main question is, how does this predict human efficacy? And we got really disappointed with this because you can see GLP-1, which is the really efficacious human drug, works. And DNP also works, although it's not safe. So while it gives weight loss, it's not a safe drug to take. Whereas on the other hand, drugs like FGF21 agonists cause weight loss at both temperatures, but does not produce clinically meaningful weight loss in humans. So unfortunately, the conclusion from this work is that 22 and 30 neither improves the prediction of a human obesity drug efficacy. So I think this is a sort of emperor has no clothes slide. I think it's pretty definitive at this point that we don't know how to optimize based on ambient temperature mouse physiology for prediction of human obesity drug efficacy. So the next thing we wanted to look at was what happens at the upper end of the thermoneutral zone. And it's easy to think about here in this where you're looking at energy consumption by a city, where you see when it's a thermoneutral zone for people, below it, you turn on your heat and expend energy. And again, this is why this has to be a straight line, because you're not going to turn up the heat way hot and open a window. You just want to turn up the heat just enough to defend the temperature you want. And similarly, here you turn up the air conditioning to defend the temperature you want. But really, there's not a theoretical basis for energy expenditure with cooling in mice, and we were curious about investigating this further. So what's happening in mice? And furthermore, people had some studies had approached what happens at the upper end of the thermoneutral zone, but nobody had ever measured core body temperature while doing it. And so we did this and got our first surprise, because when you look in the dark phase, you find that energy expenditure decreases, but rather than hitting a flat thermoneutral zone, it then increased at a certain point. And that certain point turned out to be the point at which body temperature increased. So this led us to define something called the thermoneutral point, which we define as the discrete ambient temperature below which energy expenditure increases and above which core body temperature increases. And in the dark or active phase, mice have a thermoneutral point in the dark of about 33 degrees. And this is their dark phase thermoneutral zone, so the thermoneutral zone is only a point. Then we looked in the light phase and saw a different curve. We saw the sort of more standard Scholander curve, and we did have what appeared to be a thermoneutral zone. However, when you look at core body temperature, you see that what happened was core body temperature, again, is maintained at a lower temperature in the light phase. And then at some point where you let the core body temperature increase while not increasing the energy expenditure. And then finally, what happens is you reach this point, which is actually the same as the dark phase thermoneutral point. And that's the point above which core temperature increases, energy expenditure increases, because the animal's just lost control of its thermal biology. So the point is, in the light or resting phase, mice have a thermoneutral point light, and a second break point, that's a thermoneutral point dark. And humans have a thermoneutral zone spanning 10 degrees. They do not have a thermoneutral point. This is just a fundamental difference in the thermal physiology between humans and large mammals and small mammals. And I think this is going to, I think this has not been studied in detail for many species, but I think this is going to be a general physiology principle. So what are the implications for this for studying mice? Everybody wants to study mice at thermoneutrality, But I think this data means that studying mice at thermoneutrality is really not possible. And one reason is that when you're increasing body temperature in that light phase, you're increasing the inflammatory immune tone. So you're potentially increasing immune tone by studying mice at, say, 30 degrees, which is what people have used as thermoneutrality in the past. And others have suggested other temperatures for studying mice. We decided on suggesting a temperature of 28 to 29. And the rationale for that is you want to stay below the thermoneutral point at all times to avoid the body temperature increases. Yet you want to stay close to the thermoneutral point to minimize cold-induced thermogenesis. And this allows the mouse to use its usual thermal regulation because mice live below thermoneutrality. Thermoneutrality is not where they're used to hanging out. So that brings up the question, what is the ambient temperature most predictive of human physiology? And I would argue that there is no best temperature. And there's no simple answer. The choice depends on the purpose of your experiment. You got to think it through for each experiment. And I would argue that 30 degrees is not thermoneutrality. It is 30 degrees. And I would suggest instead of calling it thermoneutrality, just call it 30 degrees and describe what it really is. Okay. The next point I want to bring up is, what about all the fine structure? I sort of ignored a lot of the fine structure in the data. But you see, there's all this information here. What's really going on? And so what people have done is said, well, there's a lot of physical. This is a human example from Bobo Gardner's Eric Ravison from a bunch of years ago, where they said, well, what is the cost of physical activity? They extrapolate human physical activity to zero. And from this line, they can now get a measure of human physical activity energy expenditure. We and others have applied this to mice, but I now think this is not the right thing to do. And why is that? Because of this. This is looking at an example of a single mouse studied at 22 degrees in the light phase, dark phase, where we've measured energy expenditure in the black, body temperature in the blue, physical activity in the orange, and food intake and water intake in the pink. And what you see is these are incredibly well correlated. And so what Voitoscope did was to set a computer algorithm to identify these bouts of increased energy expenditure based only on the energy expenditure, and they colored them in with this gray color. And these physical activity, these energy expenditure bouts contain the vast majority of physical activity, of food intake, of water intake, and they have increased body temperature. And for all, and then we did EEGs and EMGs and showed that these bouts are really episodes of active awake. And so for all parameters, the light-dark phase correlated less well than bout status did. And the metric that correlated best with light-dark was body temperature, and light-dark explained 0.67 degrees difference in temperature, but between bout explained 0.88 degrees of temperature. So bout status rather than light-dark phase is a better way of understanding physiology in mice. And then that reads the question, so what is driving? So what is driving this increase in energy expenditure? Is it the physical activity? Is physical activity increasing, driving energy expenditure and body temperature? Or what's going on here? So we came up with two models. One is that physical activity, the heat from muscle work is doing it. So muscle is doing work, increasing energy expenditure, generating heat. And since muscle is only about 20% efficient, so of all the mechanical production, there's another 80% of energy production and thus heat. And so maybe the heat from muscles is increasing temperature, increasing energy expenditure, and that would indicate a reduced need from brown adipose thermogenesis. Alternatively, you say maybe there's some central awake active state signal that's characterized, that signals a higher defended body temperature, which will increase thermogenesis to drive the warmer body temperature and will drive physical activity. And there may be an increased need for bat thermogenesis. And so we had 3 tests of this, and all 3 tests supported the arousal model rather than the heat from muscle work model. And those tests were, 1, measuring the actual cost of physical activity energy expenditure. We did this by doing this at a warm enough temperature that you weren't neutralizing cold-induced thermogenesis, and therefore you're really measuring the full effect of physical activity. And it turns out physical activity explains less than half of the energy expenditure increase during these arousal bouts. The second thing is if physical activity is causing heat, it should reduce brown fat temperature, whereas if you have arousal and arousal wants a higher body temperature, you should see an increase in brown fat. And indeed, you see a brown fat temperature increase. It actually drives and precedes the increase in core body temperature. And this led us to a hypothesis that maybe energy for body temperature regulation is really what's causing a lot of this physiology. And I'm going to show you next that indeed energy expenditure to drive body temperature really can explain mouse energy homeostasis. And I'm going to show just a little bit of that. And so one of the ways we explored that is said, okay, does a change in body temperature predict changes in energy expenditure? And so we just did, and I took out all the math, but we just said, okay, there's a change in heat content, which is simply due to the change in heat produced minus heat loss. And when you do that, you make a model, the only inputs to the model, the only varying inputs were the body temperature time course and physical activity. And what physical activity was used as is a binary indicator of bout versus between bout. It wasn't used for the energy content of the physical activity. And the reason it's important to have bout versus between bout is it turns out that there's a state change in the energy physiology. During a bout, you change the body conductance, and the bout and between bout conductances are different. So the model output was based on the body temperature time course. You determine an energy expenditure time course, and the physical activity allows you to fit it with two different heat conductances. And since one picture is worth 1,000 words, here's one example of the model. Here's an example of that same mouse, actually, just to keep your eyes. This is the measured body temperature. This is the measured energy expenditure in black, and the model-derived energy expenditure in red, and the measured heat conductance in black, and the physical activity generated heat conductance in red. So you see that this is, this is one mouse, one day of one mouse. We did multiple days of 23 mice, and the overall correlation coefficient of the measured energy expenditure and calculated model-derived energy expenditure based basically totally on body temperature was 80%, was 0.80, 80% of the variance is explained. So to summarize, what this says is what happens is mice have these bouts of active awake state, which drive a higher defended body temperature, increasing energy expenditure. They have a higher conductance within the bouts, a higher heart rate, higher blood pressure, virtually all activity, food and water intake. Compared to these resting or sleeping bouts, where you have a lower temperature, a lower energy expenditure, a lower conductance, lower heart rate, lower blood pressure. And there's on the order of 5 bouts per each 12-hour light and dark phase. So to summarize, bout status correlates better with physiology than does light-dark phase. And we propose that defended body temperature is what's driving energy expenditure changes, dependent on the centrally determined defended body temperature changes. And again, just parenthetically, the defense of the lower body temperature is completely active. This is not a passive consequence of resting and sleeping and uncontrolled heat loss. So is this useful or anything? So I've given you this bout model. We're about the time when we were writing this up, and I don't think we'd submitted it yet, or maybe even submitted the first journal that it was rejected at. And we saw a paper from Alex Banks in Molecular Metabolism, where he had done 24-hour continuous glucose monitoring in mice and also energy expenditure. And we looked at his data and go, my, that looks incredibly correlated. And when you do this overlapping of his two graphs, you see that indeed bout status correlates in as determined by energy expenditure here, also correlates with glucose levels. Again, a difference in mouse physiology, but from human physiology that I think really was appreciated before Alex's data. And we would add that basically this is a sleeping versus awake change in glucose at an oversimplified level. Um, so I want to close. Yeah, I'm talking too fast. I want to close a little bit about the relationship in body temperature and energy expenditure in mice compared to humans. In mice, body temperature drives energy expenditure. A higher body temperature requires more energy expenditure, and that's a consequence of mice living below thermoneutrality and having such great amounts of heat loss. In humans, body temperature does not drive energy expenditure. Body temperature is regulated without the need for energy expenditure changes. Since humans live in thermoneutrality, we adjust our clothes, we adjust our thermostats. But body temperature does affect metabolic rate in humans. And this is information that I think is sort of lost. So I'm trying to be an advocate for its resurrection. This is data from Dubois 100 years ago showing that human body temperature does affect metabolic rate, and it affects about an 8% increase in metabolic rate per degree Celsius increase in core body temperature. And we re-examined this independently, applying a correction factor of 2.3, which I won't go into now, but you can read about it if you want, in data from Ansel Keys with lots of data. And indeed, we found that correcting his resting basal metabolic rates improved the coefficient of variance. And we've suggested that when reporting basal metabolic rate, one should also report the basal metabolic rate adjusted to a reference body temperature, such as 37 degrees. And again, since I have a captive audience, I'm proselytizing here. And the last thing I want to talk about is, what are the relative importances of energy expenditure and energy intake in the treatment of obesity? And this is a collaborative study with Rune Curry's group at Novo Nordisk. I have no financial stake in any of this. So what Rune's group did was use the combination of cagrilintide, which is an amylin mimetic, and semaglutide, which is a GLP-1 mimetic, a GLP-1 agonist, and treated diet-induced obese rats with this drug, which is called CagriSema, compared it to vehicle treatment of those diet-induced obese rats, compared to rats pair-fed through the reduced food intake of the CagriSema treatment rats. And the magic of this study was actually a weight match group where rats were further underfed to match the body weight of the CagriSema treated rats. And that's shown graphically here. This was the treatment for 3 weeks. The vehicle rats gained a little bit of body weight. The CagriSema treated rats lost 12% of their body weight. However, the pair-fed group, pair-fed to the CagriSema, lost only 8% of body weight, and you had to underfeed further to weight match to the weight loss of CagriSema. And that's shown here. What were those numbers? Well, if you look at it, CagriSema reduced the 3-week food intake by 39%, and again, pair feeding was a 39% reduction. Whereas if you wanted to weight match to CagriSema, you had to have a 51% area under the curve food intake reduction. So this really is a graphical illustration that this drug is affecting both food intake and metabolic rate. And again, I showed you just basically the first and last before, but this shows you a time course of what CagriSema treatment does to food intake. And you see that it causes a massive reduction in food intake that gradually recovers, but not completely, by the end of the 3-week treatment. Whereas here's the weight match group and here's the pair-fed group overlapping CagriSema. Now, what happened to metabolic rate? Whereas this is the full scale in energy intake, metabolic rate's a truncated scale, and you see that CagriSema didn't do much to metabolic rate. It sort of really, it prevented the reduction in metabolic rate that the caloric restriction caused. So the reduction in metabolic rate caused by caloric restriction is called metabolic adaptation. And that's why dieting is so hard, because when you diet, your body goes, oh my God, this isn't good. I'm starving, or it's perceived as starving in some way. And so your body becomes more efficient. And that's what these two curves are showing you. If you underfeed more, metabolic rate drops more. If you underfeed less, metabolic rate drops, but drops less. But the point is, this is the weight matched group to this. So there's really a big difference in metabolic rate between the caloric restriction group and the non-caloric and the drug treated group. And finally, just I like RER because it sort of tells you have you really reached steady state or not, meaning you should be mimicking your food quotient as indicated by the vehicle group. And you see we're pretty close here, whether it's exactly steady state or not, but we're close to steady state at 3 weeks of treatment. So then I just want to take you through a little further analysis of the data. This is, again, the same first graph, now just with energy intake effect identified as the difference between the pair-fed group and the vehicle group, whereas the CagriSema group versus the pair-fed group, you can now, this is an energy expenditure effect, the lack of metabolic adaptation. And I'm not going to perseverate over it because you're subtracting big numbers here, but basically, by the steady state level, by about 3 weeks, slightly over 1/3 of the weight loss effect was due to energy expenditure, lack of metabolic adaptation, where slightly less than 2/3 of the drug effect was due to an energy intake effect. And so now what we're going to do is analyze the data comparing baseline to the 3-week data points. And the first thing we did was show the change in body weight and the change in energy expenditure. Again, here are the vehicle rats. They bounce. And I'm sorry, let me set this up a little better. This is data from 145 individual rats pooled from 15 experiments where all the vehicle rats were treated with vehicle. We had a drug-treated group with rats either with CagriSema, with Cagri alone, or Sema alone, and we had a caloric restriction group treated where we pooled pair-fed and weight-matched rats. And that allowed us to get us some range of efficacy and everything. And by having enough numbers, we could do some real proper analysis of covariance around this. First thing you see here is, again, you see the expected, if you're going to change body weight, you're going to try and recover from it, the expected change in energy expenditure. And what's interesting is the caloric restriction is really basically on the same line, but the drug-treated animals are on a different line. A different way of looking at this is now not at the change in energy expenditure, but just the final energy expenditure and the final body weight. And now you can see that the vehicle groups, in this case, overlap the drug-treated groups, whereas the energy expenditure is reduced. This is the metabolic adaptation you see with drug treatment that you're not seeing with CagriSema or Cagri or Sema. And finally, this is when you do the same sort of analysis on the energy intake and body weight. You see there really are 3 different rules. The vehicle guys are doing this. The caloric restriction guys are really reducing their energy intake because we forced them to. But the CagriSema, which are weight matched to this group, have not needed to reduce their energy intake enough to match the body weight. So let me summarize this CagriSema data and say that initially CagriSema severely lowers food intake, which recovers some approaching a new steady state by 3 weeks or abouts. CagriSema prevented metabolic adaptation. CagriSema treated rats do not reduce their energy expenditure, unlike when weight reduction is caused by caloric restriction. At the 3-week time point, about a little less than 2/3 of weight loss was from reduced food intake, and maybe a little more than 1/3 was from the lack of metabolic adaptation. And I didn't show you the data, but individually, Cagri and Sema also behave similar to CagriSema, meaning they prevent some metabolic adaptation. And again, this is work probably about to be accepted. It's currently in review, and it's work from Novo Nordisk. I have no financial stake in any of this. I'm a lucky collaborator who's given the data to be able to look at it. Okay, so let me summarize the talk. Environmental temperature and obesity. The thermal physiology of humans as mice is fundamentally different. There's no magic way to convert a mouse to a human thermally. Small mammals need to generate and conserve heat, whereas large mammals need to dissipate heat. Humans typically live and are studied under thermoneutral conditions. Mice are typically studied below thermoneutrality and typically live below thermoneutrality. Drugs can have different effects when studied at thermoneutrality versus below it, and there's no magic condition of ambient temperature that better predicts mouse drug efficacy for obesity. Mouse thermal physiology is better described by thermoneutral points, TNP, rather than by thermoneutral zones that work better for larger size homeotherms. And again, to reiterate our definition of TNP, the TNP is the ambient temperature below which energy expenditure increases and above which body temperature increases. And I just want to point out that the definition of the thermoneutral zone does not include consideration of body temperature. And I strongly believe that there should be a revision of the definition of the thermoneutral zone, that you do include body temperature, because I think you're not understanding the full physiology unless you also monitor body temperature for your definition, for your analysis of thermoneutral zones. And the last thing I want to say is mice, even though mice don't model human thermal physiology, understanding how mouse thermal physiology works makes them a great model. And and we just have to know that mice and human thermal physiology is different, and we have to understand those species differences. And I'd like to finish by saying I have a multi-decade collaborator, Oksana Gavrilova, who's participated in the vast majority, all the work from my lab, I will say. And a lot of the thermal physiology work was done by Wojciech Skok, who's now an independent professor in Prague. I have another long-term collaborator, Chuying Zhao. And I didn't get a chance to talk to you about some great CNS control of this by Ramon Pignol. And Gustavo did some of the work I showed you. There's also collaboration with on the human physiology with collaborators in Phoenix, and most of the drug work and all of the CagriSema work, it was done at Novo Nordisk through a team led by Rune Kure, and I've collaborated with them on the analysis of that data. And some of the modeling was done with input from Kevin Hall and Kong Chen, and I thank you very much.
Thank you so much, Dr. Raymond. This was such a wonderful presentation. So let's try to do as many questions we can. The first one in the chat said, why should indirect calorimetric data not be normalized by body weight?
Ooh, let's see, do I have extras in this one? No, I'm sorry, I don't have any extras. Okay, let me stop sharing so you can see my face.
Yes,
sorry.
We can see your face.
Oh, you can? Okay, I
Yes,
can't
yes,
see
yes.
you. Okay.
Yeah.
So I'll look at my camera. So the reason is that if you have mice that are heavier, you're dividing by a larger number. Well, the first reason is that dividing by body weight implies that the line will go through the origin, will go through zero, meaning that there's no y-intercept above zero. The y-intercept is zero. And that's simply not true. So it's mathematically incorrect to do that. And the errors you make depend greatly on whether you're comparing a mouse that's smaller, lighter, or heavier. So that's one reason. The second reason is that different tissues have different metabolic rates. So typically you're comparing obese mice or obese animals and lean animals. Well, they may have a similar-ish amount, maybe of lean body mass, which has a higher metabolic rate than the fat mass does, which is somewhat inert. So there's lots of reasons not to. You just get the wrong answer, and it's very misleading. You should report, like I did at the end, graphs of metabolic rate as a function of body weight, and analyze if your groups do differ by body weight, and analyze it by analysis of covariance.
A following up question, what about normalizing to lean body mass?
That's theoretically not a bad idea. My experience with it is by measuring, there's a measurement error in lean body mass. And body weight does a good enough job that it more than makes up for the measurement error that you get from your echo MRI or whoever you measured your lean body mass. So basically, you get extra errors of measurement that turn out to mess up the data. So theoretically, the best thing to do would be to analyze it as a function of lean body weight and fat mass, both, right? That's going to take advantage of the metabolic rate of the fat mass and the metabolic rate of the lean body weight and get the best answer. But empirically, in real world data analysis, body weight works just fine. It gives you the right answer, and it eliminates all these measurement errors that people have never done enough animals to properly account for.
Great. For studying effect of high temperature conditions during the active phase, mimicking human occupational exposure to high temperature, will you suggest using 29 Celsius during light phase and then higher than 33 in the dark phase, how we should set our temperatures in the animal facility?
Yeah, that gets really, you start getting animal welfare issues. Because if you put a mouse at 33 and then give them CL, or 35, you can do bad. They will become hyperthermic. So you're gonna have to stop the experiment. I would approach it a little differently, and if you really think you're going to be causing an increase in body temperature, I might start with 30, not go above that to say 32 or 33, and then monitor body temperature so that you're not going to do harm to the animal. Those are hard studies to do because temperature changes much faster in a small animal, and they can't handle it. And yeah, I'll stop there.
So if we don't want to study temperature conditions, we should use 29 throughout the day and night?
I like 29.
You like 29?
I like 29 because I think you're minimizing cold-induced thermogenesis, and you're not thermally stressing the animal.
Yeah. So what can you conclude from the use of mice at normal mouse house condition of 22 for decades in terms of what they are telling us? Can you comment on immunology, neurophysiology changes with the 22 temperature that we used
I
to
like
use?
22 because I
You
think
like 22?
I like 22 because it allows the mouse, as long as the mouse has adequate food, and typically mice have a little bit of nesting and stuff. A lot of my experiments are done without nesting to remove that. If you start adding nesting, then the things are a little less extreme. The mouse has even more control of its thermal environment. I really like those because a mouse at 22 is controlling its body temperature, is controlling its environment. As long as you give it enough food, it's fine. And so its body temperature is not going to drop. And so you're going to have normal immune system function, and the mouse is going to regulate it normally.
Okay, so you like anything between 22 and 29 as long as we put that in the manuscript, right? As long as we disclaim the temperature.
And again, the advantage of 22 is it's easy, right?
Yeah.
That's what our animal facilities are. And I really think, people do all sorts of crazy things to try and get a controlled temperature. And in reality, the temperatures are not as controlled as people think they are when you start putting thermometers in individual cages. It's really kind of amazing, actually, unfortunately. So I'm happy, depending what you're trying to do, but I'm very comfortable with studies at 22 degrees.
There are so many questions and so many things for this wonderful talk. Okay, let's do sex difference. Any data with sex difference, pregnancy? Tell us something about it.
Oh, sure. So lots of subtle differences. And it gets hard because you have to start doing 12 mice of each sex to see them properly. Okay, female mice have a higher body temperature. Let's see. I always get this mixed up. We've shown that female and male mice respond to fasting. They both go into torpor, but I forget, one does more episodes of torpor and one does fewer episodes but longer duration. And we found that, not expecting any sex difference in torpor, we found that. There's all sorts of subtle differences like that. There's differences when you start doing group housing. But even if you put related males together, they will huddle, even though males aren't supposed to like to be with each other and can fight, but they will sleep huddled. There's all sorts of stuff on sex differences. The problem is it's not particularly well documented, and it takes large numbers of animals to really have the power to show the small differences.
Yeah, because most of these study we have done in single house, right? Conditions, they are by themselves. So
Yes.
males don't like to be with each other. What about females? They do better if they are in a group?
Well, in our experience, we did that. We did a study. Voida did a study where we had 3 males compared to 3 females compared to 1 male, 1 female, and you know, multiple animals per cage reduces their food intake need and energy expenditure. Right? They huddle whether they're male or female, but the females huddle more than the males, for example.
Hmm. Okay, so a question about the drug Kagisema. So do you know if this drug, the GLP-1, affect cardiovascular parameters, basal heart rate variability, blood pressure?
Um, we have not studied that.
Okay.
Sorry about that.
Um, so let me do another one. How does the model that defend temperature TV drive TEE changes translate to torpor?
Huh? We haven't done that. I would assume it would still work, but we haven't tested it. The model was remarkably robust. What Voida did was he actually spent, I didn't get into it. We actually derived a general model of metabolic rate where we had, thermic effect of food
Mhm.
quantitated, where we had basal metabolic rate quantitated and adjusted for body weight and composite. We had done all those things. So we have a general model of metabolic rate, but we haven't tested it in that way. So the answer is I don't know on the model.
Thank you. So can we talk more about the relationship between basal metabolic rate and body temperature in the context of hypothermia, human and mice, instead of a core body temperature increase? So what happens if we lower the temperature?
Right. So the parameter for that is called a Q10 or Arrhenius constant, and a Q10 is just the change in a temperature, say a core body temperature, for a 10-degree change in core body temperature. Sorry, the change in rate for a
Mhm.
for a 10-degree change in core body. The Q10 is 10-degree change and the rate. So people typically talk about 3 as the number, but actually, if you look at the literature, 2.3 is a better number for humans and mice, I think. 2 to 3. So let's say
To
2 to 3. And so if you use 2.3, every degree centigrade is an 8% difference in metabolic rate. So if you lower body temperature by a degree, you're going to lower metabolic rate by 8%. It's a massive effect.
It's really big, right?
Yeah.
So what about for those who study drug efficacy? What is the best temperature to maintain mice if you want to show metabolism to prolong the half-life of drug being administered? Example, semaglutide.
So the changes in ambient temperature won't change drug half-life. Okay.
Okay.
Those are, again, because core body temperature, metabolic rate, so core body temperature is not really changing. And I don't, in a prior career, I worked at Merck for 9 years, so I know a fair amount of pharmacokinetics and metabolism. I would assume, although we haven't actually done that experiment, that none of these ambient temperature changes are going to change pharmacokinetics of drugs because the core body temperature is going to be pretty darn similar, and it's going to be small differences, if anything.
Thank you. So I have a question. As an endocrinologist, I see all this exciting data in a preclinical research, and then we go to the clinic and we just, get the BMI. So how we can translate these great measurements from the lab to the clinic to assess basal metabolic rate in humans?
Yeah, so one of my colleagues here, Kong Chen, the honest is we're fortunate to have indirect calorimetry chambers for people. We have 3 chambers, and Kong Chen runs our facility, and he and Rob Brickta have done a Scholander type experiment with humans, and that was some of the data I sort of had referenced on one of my slides as comparing human versus the physiology in humans and mice. Um, I One of
Mhm.
my um, hypotheses is that a successful obesity drug therapy is going to affect food intake, because food intake is the bigger contributor. But I think metabolic rate, either just preventing metabolic adaptation or having an effect on metabolic rate, is going to be important for efficacious human obesity therapies. Comparing mouse and human, probably food intake is more important in humans than mice. So I don't know if our number of 1/3 of semaglutides is the metabolic rate, if that will translate to human. Probably not. But I believe, but there's no human data for sema, that there will be a metabolic rate effect. I can't believe physiology is going to be that different.
There are
Um,
patients that they don't lose weight on GLP-1. Do you think that that is due to lack of effect in the metabolism rate or resistance in the food intake area?
I think that's a really interesting subject to study. I mean, let's say 20% of people don't tolerate GLP-1 mimetics or
Yeah.
agonists because of GI discomfort. So so those people, I think they didn't get the drug, they can't tolerate the drug. I'd be really curious if there really are people who really tolerate the drug and can take it and don't lose weight. I think those would be fascinating people to study. And
And
again, I would argue that you'd want to study them in a domiciled environment pre and post, so you really know exactly what their food intake is. Because in my humble opinion, food intake questionnaires are not worth anything. They're just not sensitive enough.
okay, we are running out of time. Thank you so much, Dr. Reitman. This was an amazing presentation. I want to thank also APS, Jacob Wright. He is on the call. Thank you so much for putting this together and all what you do at APS. And finally, I want to big thank again to our sponsor today, Columbus Instrument. Thank you so much. With your support, without your support, we are not going to be able to do many things that we do for our sessions. So I really appreciate all the help and support. And to all of you for being here today and participate of this wonderful presentation. Dr. Rainman, thank you so much again. I hope to see you soon.
Thank you very much. My
Have a
pleasure.
great day. Bye-bye.